final seminar from Alexander Birbrair. Alexander graduated from the Santa Cruz State University from Brazil in 2008 in biomedicine, and he moved to US to join our laboratory in 2009. Since then, in the last 5 years, he scored several goals like we are in soccer time. So he put 9 goals in total, so 3 assists, and he has 2 papers under review. So probably he's going to complete his experience with us in publishing around 12 papers, something that is really amazing. So his work is based on the role of PERICYTE. It's a very hot topic, but it's also under debate in many areas. So that's why I really encourage you, especially those that are not in the committee, to make questions. Because that's going to help him, because pericytes are in the vasculature in every tissue, and therefore they may influence not only the biology of the tissue, not only the capacity of the tissue to be repaired, but it also will influence other stem cells, because pericytes form the niche for stem cells in many organs.
Thank you. Good morning, everybody. Thanks a lot, everybody, for being here today. So the title of my talk will be Pericytes at the intersection between tissue regeneration and pathology. It is widely accepted that tissue regeneration depends on stem cells. However, under certain pathological conditions, those cells may form some unwanted tissues, such as fibrous and adipose tissue, which impair organ function. What we found here is that sometimes the pericytes that form those undesired tissues differ from the ones that participate in tissue regeneration. So initially, I will introduce to you the concept of pericytes functioning as stem cells. Every tissue has its own committed stem cells which are restricted to form cells of this specific tissue. This is, for instance, happening in the central nervous system with the neural stem cells, in the skin with the skin-specific stem cells, in the muscle with the satellite cells. There are also other stem cells that are not tissue-restricted, the mesenchymal stem cells. They are multipotent, being able to form a variety of tissues. They contribute to tissue cell turnover and respond to damage. Their precise locations are unclear, and they are present in small numbers. Because of their broad distribution in all organs, it has been proposed that maybe they can have a common origin. As blood vessels are also present in almost every tissue in the body, cells associated with the blood vessels, called pericytes, have been proposed to be mesenchymal stem cells. Pericytes are characterized by their anatomical location. They are in a very close proximity to blood vessel endothelial cells. Their main function is related with vascular stability. So in this image, by analogy, this will be the pericytes which stabilize the vessels. They have multiple other functions. Here we will focus specifically on their function as multipotent stem cells. Pericytes can form muscle, cartilage, adipose tissue, and bone. And now I will show you that in skeletal muscle cultures, pericytes can form neural progenitors. In our recent work, we isolated neural progenitors from skeletal muscle cultures. For this, we used a transgenic mice that is called nestin GFP, in which the GFP fluorescence is under control of nestin regulatory elements. Nestin is a cytoskeletal protein which is a marker of multiple stem cells, including neural stem cells. When we extract the muscles from those animals and culture them under optimized culture conditions, after 10 days, all the cells that express nestin GFP fluorescence also co-express multiple neural progenitor markers, for instance, TUJ1. Those cells also had neural morphology. As neural progenitors, are known to respond to neurotransmitters, we tested whether those cells could respond to neurotransmitters. So we did whole cell voltage clamping those cells and exposed them to short pulses of glutamate. And we observed that the cells responded with typical inward currents, while when we exposed those cells for a little bit longer to glutamate, in this case for 1 second, those cells stopped responding, as some of the glutamate receptors have the characteristic to desensitize when exposed for a longer time to the agonist. In addition, we injected those cells in the brain striatum, as shown here in the green dot. Those cells survived and migrated to the subventricular zone, which is a very special area located around the lateral ventricle, which is the natural neurogenic area where the endogenous neural stem cells are located. Those cells were very similar to a specific type of neural progenitors, which are called Ng2 glia cells, as by immunocytochemistry they expressed multiple markers of those cells, like oligodendrocyte progenitor marker 4, platelet derived growth factor receptor alpha, nerve growth factor receptor, and the glutamate receptor AMPA. Notice that this result is in agreement with our electrophysiological data, as the glutamate receptor AMPA has the characteristic to desensitize when exposed for a longer time to glutamate. Those cells had outward potassium currents and expressed the potassium channels KIR 6.1, specific of Ng2 glia cells. When we cultured those cells in dishes uncoated, they formed these floating neurospheres, which, when seeded into dishes pre-coated with laminin, attach, forming those monolayers in which the cells made contacts between them, which are additional characteristics of neural progenitors. So based on our findings, we hypothesized that we can collect a skeletal muscle biopsy, culture the cells under optimized culture conditions, and by sorting based on surface markers, we could isolate pure neural progenitors, which could be used for cell therapy approaches, what we are currently testing in our lab. However, those neural progenitors, they are not present in the intact skeletal muscle as such. They are formed after culture. So our next question was, what cells in the skeletal muscle have the potential to originate those neural progenitors after culture? As in the central nervous system, pericytes can be induced to differentiate into the neural lineage, we hypothesized that pericytes from the skeletal muscle also could have this potential. So then we crossed our nesting GFP mice with Ng2DS red mice, in which all the pericytes which express Ng2 proteoglycan are marked by DS red fluorescence. Then, by immunohistochemistry, we analyzed the skeletal muscle of the double transgenic animals obtained. So in the skeletal muscles of those mice, we found 2 different populations of Ng2DS red positive cells. Some cells that do not express nesting GFP fluorescence, but also express some other pericytic markers as platelet derived growth factor receptor beta and CD146, and another populations of Ng2DS red positive cells that do express nesting GFP fluorescence and also express the other pericytic markers. Confirming their pericytic nature, we found that both cell populations are located in a very close proximity to CD31 blood vessel endothelial cells. So in summary, we found 2 different pericyte subtypes in the skeletal muscle. We called type 1 pericytes from now on the ones that do not express nesting GFP fluorescence, while we called type 2 pericytes the ones that express nesting GFP fluorescence. Next, from the skeletal muscle of those double transgenic mice, we isolated by sorting based on GFP and DS red fluorescence 4 different cell populations and cultured them separately for 10 days. After this time, we found that no of these cell populations form nesting GFP positive neural progenitors in our culture conditions. Then we hypothesized that probably the cell population in those skeletal muscle cultures that may originate neural progenitors need some secreted factors or contacts from the other cells present in those skeletal muscle cultures. To address this point, we co-culture again each of these cell populations from the transgenic mice together with a whole skeletal muscle culture from wild-type animals. And we found that only one of these cell populations can form nesting GFP-positive neural progenitors after 10 days. That was the cell population that expressed nestin NNG2, that corresponded to type 2 pericytes. When we cultured the pericyte subpopulations in a medium without any special supplements, both pericyte subtypes become smooth muscle cells, the reported fate of pericytes in culture. In summary, the skeletal muscle has multiple cell types. Here we outline 3 of those types. In red are the satellite cells, the muscle-specific stem cells. And in yellow and green are type 1 and type 2 pericytes. The satellite cells are committed to the myogenesis, while type 2 pericytes in green, they are the ones that express nesting GFP fluorescence, and under optimized culture conditions, they may form those neural progenitors. While type 1 pericytes in yellow do not express nesting GFP, and under the same culture conditions, they only form smooth muscle cells. Our results suggest that the 2 pericyte subtypes in the skeletal muscle differ in their differentiation potential, as pericytes have been shown to participate also in skeletal muscle regeneration. Our next question was whether the role of pericyte subtypes differ also in skeletal muscle regeneration. So in the next part of the talk, I will be speaking to you about the role of the pericyte subpopulations in skeletal muscle regeneration. In the diseased skeletal muscle, fibrous and adipose tissue accumulate, affecting skeletal muscle function and its regenerative capacity. So for instance, this is a slice from a patient with muscular dystrophy. Most of the skeletal muscle is replaced by fat tissue shown here in white. This has been associated with changes in stem cell function. The most well-studied stem cells in the skeletal muscle are the satellite cells, which are located beneath the basal lamina of the muscle fibers, and they are committed to the muscle lineage. Besides satellite cells, there are multiple other cell types in the skeletal muscle. For instance, fibroblasts, pericytes and endothelial cells around the blood vessels, adipocytes, Schwann cells at the nerve terminals. While pericytes are multipotent, satellite cells are restricted to the myogenic lineage and cannot form other cell types. So here we hypothesize that the pericyte subpopulations play an important and distinctive role in skeletal muscle regeneration. And our preliminary results suggested that while one pericyte subtype is adipogenic and fibrogenic, the other was myogenic and vasculogenic. And our aim was to establish the regenerative capacity of the pericyte subpopulations. So initially, from the skeletal muscle of those double transgenic mice, by sorting, we isolated type 1 and type 2 pericytes. Type 1 pericytes, remember, the ones that do not express nesting GFP, but do express NG2, DS-RET, and other pericytic markers, while type 2 pericytes, the one that express both nesting GFP and NG2, DS-RET, and express other pericytic markers as well. And we found that under culture in myogenic conditions, only type 2 pericytes were able to form myotubes, these multinucleated muscle cells which stain positive to myosin heavy chain. While under adipogenic conditions, only type 1 pericytes form adipocytes, which stain positive to perilipin, that is a protein coating lipid droplets in fat cells. When we culture type 1 and type 2 pericytes under fibrogenic conditions, only type 1 pericytes start expressing the fibroblast marker FSP1 in vitro, and also they produce type 1 collagen, suggesting that in summary, in vitro, type 1 pericytes, the ones that do not express nesting GFP fluorescence, are adipogenic and fibrogenic, while type 2 pericytes, the ones that express nesting GFP, are myogenic. Now, how to track the fate of type 1 and type 2 pericytes in vivo? To address this point, we made use of beta-actin DS-RET mice, which we crossed with our nesting GFP mice, and in the double transgenic mice obtained, all the cells had DS-RET fluorescence. Then we isolated the pericyte subpopulations from the skeletal muscle of those double transgenic mice by sorting using NG2 APC antibody, And now remember, both type 1 and type 2 pericytes have DS red fluorescence, enabling us to track their fate even after injection into another animals. To study fat formation in the skeletal muscle, we used a classical model of adipose tissue accumulation in the muscle induced by glycerol injection. Then we transplanted either type 1 or type 2 DS red positive pericytes, and after 2 weeks, we removed the muscles from those animals for analysis. In the muscles that were injected with type 1 pericytes, we found that perilipin positive cells also express DS red fluorescence, meaning that type 1 pericytes can start expressing perilipin also in vivo. While in the muscles that were injected with type 2 pericytes, we didn't detect any overlap of perilipin with DS red fluorescence, indicating that only type 1 pericytes are adipogenic also in vivo. Next, to study normal muscle formation after injury, we transplanted either type 1 or type 2 DS red positive pericytes into a classical mouse model of muscle injury induced by barium chloride. And after 2 weeks, we found that only muscles that were injected with type 2 pericytes had those DS red positive myofibers. We confirmed our findings in sections of those muscles. And we can see here that while type 1 pericytes remain in the interstitial space of the skeletal muscle, type 2 pericytes were able to form DS red positive myofibers with central nuclei, suggesting that only type 2 pericytes are myogenic also in vivo. So in summary, type 2 pericytes may contribute to the muscle formation after injury in the healthy skeletal muscle, while type 1 pericytes may contribute to the adipose tissue accumulation that is happening in the diseased skeletal muscle, as for instance in muscular dystrophy, when most of the skeletal muscle is replaced by fat tissue. So it is well known that aging may affect the tissue microenvironment, and specifically in the skeletal muscle after injury, fibrous tissue is formed instead of muscle. So our next question was whether the myogenic potential of type 2 pericytes can be affected by the aging microenvironment. To address this point, we injected the same number of type 2 pericytes with DS red fluorescence into injured skeletal muscles of young, middle-aged, and old mice. And after 2 weeks, we found that the number of DS red positive myofibers derived from type 2 pericytes decreased in the old mice. And also the cross-sectional area of those myofibers derived from DS red positive type 2 pericytes as well decreased with aging, suggesting that the microenvironment may affect the myogenic potential of type 2 pericytes. In addition, we injected type 1 pericyte with DS red fluorescence into injured skeletal muscles of old mice. And we found that differently than type 2 pericytes, after 2 weeks, type 1 pericytes didn't form DS red positive myofibers. Instead, they remained in the interstitial space of the skeletal muscle, producing type 1 collagen, and also they expressed the fibroblast marker FSP1, indicating that only type 1 pericytes are fibrogenic in the old muscle in vivo. So in summary, in the healthy skeletal muscle after injury, type 2 pericytes, together with satellite cells, may contribute to the muscle formation. While in the old skeletal muscle after injury, type 2 pericytes also may contribute to muscle formation. However, those muscle fibers are smaller and fewer, while type 1 pericytes contribute to the fibrous tissue formation that is happening in the old skeletal muscle after injury. Now, in the next part of the talk, I will be speaking to you about some additional roles that we found of pericyte subtypes, as for instance in fibrosis in other tissues. So first, we administered the fibrogenic agent bleomycin in the lungs of nesting GFP and G2 DS red transgenic mice. And after 2 weeks, this results in lung fibrosis. In those mice, we found that the number of type 1 pericytes increased significantly. They corresponded to approximately 15% of the collagen-producing cells, while type 2 pericytes didn't generate any detectable collagen, suggesting that type 1 pericytes accumulate at the injury site after lung injury and contribute to pulmonary fibrosis. Next, we induced unilateral urethral obstruction in nesting GFP and G2DS rat transgenic mice, and after 2 weeks this generates renal fibrosis. In those mice also, we found that the number of type 1 pericytes increased significantly after renal injury, while type 2 pericytes did not. Also, we detected a high concentration of those type 1 pericytes near areas dense in collagen accumulation. However, those type 1 pericytes were not producing any type 1 collagen, suggesting that type 1 pericytes do not contribute to kidney fibrosis. To verify whether pericytes participate in the fibrosis formation after myocardial injury, we induced infarct in nesting GFP and G2DS rat transgenic mice by ligating the lateral anterior descending coronary artery, what resulted after 2 weeks in myocardium with dense scar tissue accumulation. In those mice, we found that the number of type 1 pericytes increased significantly at the infarcted area. However, they did not produce type 1 collagen, which also accumulated in the same infarcted area, suggesting that type 1 pericytes are recruited to the infarcted location after myocardial injury, but they do not contribute to tissue fibrosis. To verify whether pericytes can participate in the scar formation after spinal cord injury, we performed a cut in the dorsal funiculus of nesting GFP and G2DS rat transgenic mice, and after 2 weeks we found the type 1 pericytes, the ones that express NG2DS rat but do not express nesting GFP fluorescence, they increase significantly at the local where the scar is formed after spinal cord injury, while the cells that are positive for NG2 and nestin distributed throughout the spinal cord section. In addition, we performed a similar injury in one of the sides of the brain of the nesting GFP and G2DS rat transgenic mice. And after 2 weeks, we found that in the injured side of the brain, in the scar tissue, type 1 pericytes accumulated in comparison with the contralateral uninjured side of the brain. However, those NG2DS rat positive pericytes didn't overlap with PDGFR-beta positive cells, as PDGFR-beta is a marker of several other cell types, such as fibroblasts. Our results suggest that PDGFR-beta positive cells, which are functionally contributing to central nervous system fibrosis, differ from the NG2DS rat positive pericytes in the central nervous system. So in summary, our results suggest that type 1 pericytes accumulate after tissue injury. However, they only produce collagen in an organ-dependent manner. Now I will speak to you about the roles of pericyte subtypes in angiogenesis, vessel formation. Both endothelial cells and pericytes are needed for vessel formation. So our next question was whether both pericyte subtypes are angiogenic. To test their angiogenic capacity in vitro, we made use of human umbilical vein endothelial cells, which we co-cultured together with isolated either type 1 or type 2 DS rat positive pericytes. After co-culture with type 1 pericytes, we didn't detect any vessel formation, while when co-culture with type 2 pericytes, we found structures very similar to blood vessels in vitro, suggesting that only type 2 pericytes, surprisingly, are angiogenic in vitro. To confirm their angiogenic potential also in vivo, we added to the mixture of uvic cells plus either type 1 or type 2 DS rat positive pericytes matrigel, which induces vessel formation. And then we injected all this mixture subcutaneously into nude mice, and we recovered the matrigel plaques for analysis after 2 weeks. We found that while in the plaques that were injected with type 1 pericytes we didn't detect any vessels, in the plaques that were injected with type 2 pericytes, now they were vascularized. And in addition, we detected beta arcs in DS red fluorescence around the blood vessels in the plaques that were injected with type 2 pericytes, indicating that only type 2 pericytes are angiogenic also in vivo. The tumors, when they grow, they form new blood vessels, which recruit cells from the host. So as forming new blood vessels is the process that is called angiogenesis, our next question was whether both pericyte subtypes are recruited during tumor angiogenesis. Here I'm showing you a section of one of those brain. These lines separate the normal brain tissue from the tumoral tissue. So in the normal brain, we found two different types of Ng2-DS red positive cells, as shown in the red box, cells that express Ng2-DS red fluorescence but do not express nestin GFP. While in the yellow box, in the normal brain tissue, we found also cells that express Ng2-DS red fluorescence and nestin GFP fluorescence. While invading the tumors, as shown for instance in the white box, we only found cells that express both Ng2-DS red and nestin GFP. However, in the central nervous system, besides type 2 pericytes, oligodendrocytes progenitors also express Ng2-DS red and nestin GFP. As oligodendrocytes progenitors are not present in the periphery, we injected tumor cells subcutaneously into nestin GFP Ng2-DS red transgenic mice. And approximately after 2 weeks, we removed the tumoral tissue together with its surrounding normal tissue for analysis. And by immunohistochemistry, we can see here that in the normal tissue, in this case skeletal muscle, we found two different types of pericytes. Type 1 pericytes that express Ng2-DS red fluorescence do not express nestin GFP fluorescence and are located very close to CD31 blood vessel endothelial cells. And in the yellow box, we also found type 2 pericytes that express both Ng2-DS red and nestin GFP and also are located close to blood vessel endothelial cells. While in the tumoral blood vessels, marked here with CD31, we only detected type 2 pericytes, indicating that only type 2 pericytes are recruited during tumor angiogenesis. Now, could the type 2 pericytes also be used for therapeutic angiogenesis? The stimulation of angiogenesis may improve the blood perfusion and the function in ischemic tissues. For instance, a condition called critical limb ischemia is caused by blood vessel obstruction and constriction, which may lead to limb necrosis and even amputation in some cases. So we used a mouse model of this condition, critical limb ischemia, in which we ligate and cut the femoral artery in those mice, what stops the blood flow in those limbs, making the limbs ischemic. Then we analyze those animals by MRI angiography. We can see that in the contralateral unaffected limb, we can detect blood flow in the femoral artery, while in the ischemic limb, this blood flow disappears. Then we injected either type 1 or type 2 DS red positive pericytes. And after 2 weeks, we analyzed by the same technique again the limbs of those animals. We found that while type 1 pericytes were unable to recover the blood flow in this artery, type 2 pericytes recovered the blood flow in the femoral artery after 2 weeks. Now I will show you the same results in three-dimensional images of the limbs of those animals below the knee. So we can see that while type 1 pericytes didn't recover the blood flow in the femoral artery, type 2 pericytes were able to partially recover the blood flow in this artery. In addition, by immunohistochemistry, we detected the type 2 pericytes surrounding the newly formed blood vessels in the limbs of those animals injected with type 2 pericytes, indicating that type 2 pericytes could be used also for therapeutic angiogenesis. Now, in a general summary of our story, the pericytes as a whole population, they are multipotent. What we found here is that pericytes are heterogeneous, and there is at least 2 pericyte subpopulations, which are oligopotent. They are more lineage restricted,
While type 1 pericytes may form fat and fibrous tissue, type 2 pericytes may form muscle, neural cells, and blood vessels. In conclusion, while one pericyte subtypes favor vasculogenesis and myogenesis, the other counterbalances these positive outcomes by forming fibrous and adipose tissue. Also, we have shown that type 1 pericytes' participation in fibrosis is tissue dependent. And also, we demonstrate that only type 2 pericytes are recruited during tumor angiogenesis. Finally, our model provides central cellular targets susceptible in the future to signaling manipulation and pharmacological manipulation. And with this, I would like to thank several people without which nothing of this work will be done. So all this I will read because I didn't memorize. So all this will not be done without multiple people. So first of all, I need to thank a lot I would like to especially thank my mentor, Dr. Osvaldo Del Bono. I was very lucky to have such a mentor, for guiding me throughout this PhD, being supportive, approachable, and always motivating me. Also, I would like to thank all Del Bono Lab. I was very lucky to be part of this wonderful group. I would like to thank the past members, as for instance Jackson, and all the current members, Laura, Tan, Somin, and Andrea. Especially I would like to thank Laura for being so supportive and patient with me. I would like to thank my co-mentor, Dr. Akiva Mintz, and his lab. He always supported me strongly and always offered helpful advice. I would like to thank all our collaborators and all the thesis committee for their time and their strong support. I would like to thank the Rabbi Plotkin and his family. In these more than 5 years, they have been my second family. And I would like to thank everybody that came today, and in special my friend Jimmy that came all the way from Brazil. And also I would like to thank my future wife, Veronica, always strongly supporting me. Thank you, and I will be happy to take any questions. Thank you.
Very nice work. In your muscle injury model, you are suggesting that the type 2 pericytes can turn into myofibers. How did you confirm that they were myofibers?
Okay, so let me check if I understood right the question. So your question is whether the type 2 pericytes, how can we confirm that they form muscle fibers in our muscle injury model? So what we did is that we isolated those pericytes from mice, that all the cells of those mice express DS-red fluorescence. So as well, when we isolate type 1 and type 2 pericytes, they will have DS-red fluorescence throughout all their life. So even when we injected them into the injured mouse model of a wild-type animal, all the DS-red positive cells after injury will be derived from the cells that we injected. As the normal wild-type mice, they don't express DS-red fluorescence. So as we can see, after injection of type 2 pericytes, we can see DS-red positive myofibers, but we cannot see DS-red positive myofibers when we injected multiple other cell types, as for instance type 1 pericytes. We suggest that they are fine.
Yeah, but how do you know they're myofibers?
We did it, so we did a myosin heavy chain stain that is specific of myofibers. This is first of all. And second of all, myofibers are multinucleated cells, so if we see a cell that has multiple nucleus, the only cell in the skeletal muscle are myofibers. So it was specific. Okay. Yes, thank you.
One question.
Osvaldo said to ask a lot of questions.
Can you try to detect pericytes number 1 and 2, type 1 and 2 in human muscles in situ?
Yes, okay, so let me see if I understood right. The question is whether type 1 pericytes and type 2 pericytes exist in the human skeletal muscle. I have two answers to your question, but they will both answer. So, in my opinion, we use in a mouse, transgenic mouse, in which the cells are just marked with fluorescence. They are not knockout. They are not anyhow different from the wild-type animals, or supposedly should be present in other species. So what we found in the transgenic mice, we understand that they are just a mouse model to mark the cells. So now to your question whether we found those cells in humans, how can we mark those cells in humans? As I said, the type 2 pericytes express nestin GFP fluorescence, while type 1 do not, and they both express multiple pericytes markers. So we used antibodies for this, and in sections, and we can see in wild-type mice, and we can see that the nestin antibody for the protein stain a lot more cells than the nestin transgene. So we couldn't translate it directly like this to find with nestin the specific markers of those cells. So if you give me, I have some slides to speak about this. So what we are currently doing is that we are searching for a special marker, to find a special marker in the skeletal muscle of those transgenic mice, and then after this test this marker to see if it works in the humans, because nestin was not perfect when translating into into humans. So what we did was we took type 1 and type 2 pericytes and did a microarray of each of the pericyte subtype, single RNA amplification, and then we did a whole microarray. But the problem is that we see a lot of differences. There is many genes. So there is a big difference. So where to start? So then we looked at any significant genes that we could find. We only wanted to see the significant difference. But after collecting the significant difference, here I'm showing you only the significant different genes. So it still was a lot. So then we excluded the ones that are not known to be expressed in pericyte, that they are known to be expressed some other cells. The ones that are unknown we still analyze, but if they are expressed in other cell types, it will be still not very specific to pericytes. So then after isolation of the cells, we did the microarray, so we did heat map, and then we analyzed by function all these genes. Why? Because we are specifically interested only in the ones markers that are in the membrane. Because if the marker is not in the membrane, we cannot isolate it without damaging the cells. So we only analyze the ones that are in the membrane. So even in the membrane, there is different levels of significance of the difference of those genes. So we selected only the most significant ones that are different. And it's still a lot. The ones that are plasma membrane markers, there were still a lot of genes. So all these are plasma membranes. And then what we did to confirm the gene expression, we needed to see whether the proteins are also expressed. So then we choose, first of all, we choose the ones that we could arrange to get it for free samples, because it's a big array, it's a big analysis. So we contacted some companies, the ones that we got for free, we tested them, and unfortunately, none of them was perfect to overlap exactly with one type of pericytes and do not be expressed in some other cell types. So our next step now is that we will invest into the ones that are most significantly different between the two types of pericytes that are in the plasma membrane, and also that already could have been reported in pericytes and were not reported in some other cell types. So this is the approach. So we did the same thing. So we have 83 genes to test from type 1 pericytes, and also we have 69 genes to test from type 2 pericytes. So this is what we are currently doing. I don't know if it answers a little bit. Okay, yes.
It's a really nice presentation and amazing body of work. What I found really interesting is the extent of accumulation of type 1 parasites in those fibrotic tissues. Now, you said that the production of collagen is tissue specific. Now, with this really significant accumulation of those cells, do you think that, this is just thinking, do you think they can influence production of collagen in other cells? I mean, could production of collagen be dependent on their accumulation irrespective of whether or not they produce it themselves? And I know that whether parasites produce collagen is you know, it's a controversial issue and it's different in different tissues. But is it possible that they do influence it?
So, okay, so if I understood right, your question is that we show that pericytes type 1, they accumulate in all the tissues after injury, the ones that we tested. However, not in all tissues they produce type 1 collagen. Could they have some additional functions? We do believe that they may have some additional function, as in all the tissue, for some reasons they are recruited to the injury site. They could be signaling, secreting some specific molecules. They could be transforming in some other cell types and losing the expression that we are unable to track them in this mouse model. They could also be happening. So I do agree with your hypothesis. We are.
Let's say if you don't have those cells, do you think perhaps you wouldn't have fibrosis, or it's the other cells?
So it's a very good point. So this work was done in the Anderson MD Texas. So, but they didn't analyze specific subtypes of pericytes. They analyzed the whole population of pericytes, and they did what you said. What if we take out the pericytes? So if they took out the pericytes from the tissue, they showed that also it varies in different tissue. Taking out pericytes from the skeletal muscle, the fibrosis is diminished. It's not completely ablated, but is diminished. While taking out pericytes from the kidney or lungs, or there is two different groups, and each one show controversial. They are still discussing the results. So probably because different mouse models can show different results. So in one group, they believe that pericytes do contribute to tissue fibrosis. So taking them out, they have diminished tissue fibrosis, while the other group thinks that there is no changes. But we believe that it's very varying between tissue, and what our specific focus initially was the skeletal muscle, and in the skeletal muscle seems to be, by the scientific community till now, seems to be well accepted that pericytes do participate. The next question in our work is, what is the extent of participation? Because it's known that fibroblasts and some other cells also contribute to tissue fibrosis. So we are now trying to understand and trying to see what is the contribution of each cell type. Thank you. Yeah. Osvaldo said many questions. Okay, better you. Okay.
So you said pericytes are stem cells. Those stem cells are in the vasculature and vasculature are all tissues. So, but most tissues have their own resident stem cells, like skeletal muscle has satellite cells. That is known that the one that contribute mainly to tissue repair, myogenesis. So, what is the contribution of pericytes actually in the mature tissue biology, repair or maintenance?
Okay.
How did you explore what is the relative contribution of pericyte as a stem cell versus the endogenous resident stem cell in tissue maintenance and repair?
Right. So, just to rephrase the question of Osvaldo is that pericytes are stem cells and are present in almost every tissue, as I mentioned, around the blood vessels, and they have this potential to form other tissues. But in every tissue, they also have these specific tissue-specific stem cells, which also can form this tissue. So what is the contribution of each type of stem cells and how to test it? So one of the approaches that was done with the whole pericytes population and was already done is that they removed pericytes through ablation of specific markers of pericytes. There are multiple markers that you can ablate those cells from the mice by diphtheria toxin and see how the tissue regenerates without pericytes. So they took the pericytes out, and then they see what is the level of regeneration without pericytes. So this was the pericyte contribution. Then let's take out satellite cells in the muscle, for instance. Taking out satellite cells in the muscle doesn't regenerate at all. So it could be that, like we were speaking about before, is that pericytes may contribute through some signaling or through some connection with other cells, but there is some other cells that are shown in the literature that they are very important also to regeneration. However, the question can go even deeper is how to test whether type 1 or type 2, the subtypes, and what is their contribution to tissue regeneration. We still cannot answer this question because to ablate those cells, all these mice models, they use tools, engineering tools, based on some specific markers. This was the reason that we are analyzing specific markers of the pericyte subtypes, because we still don't have one perfect only of those cells to ablate only a subpopulation. Yes, yes.
I guess the microenvironment is so key in this, and in that one study where you showed that you had to add the muscle cells to actually get the transformation. So it's dependent, as you said in your thesis, on the soluble factor. And yet also, in conjunction with that, all of your studies on the other tissues are dependent on a micro conditioned medium. How different are all of those microenvironments, and do all of them come to play a role then in the selectivity of choosing a specific cell type?
Right, right. So I didn't understand if this was a question or a comment, but let me rephrase.
A little bit of both.
So the microenvironment, the microenvironment definitely is very important. It doesn't exclude at all the endogenous capacity of the cell, because in some microenvironments, not any cell can become what we want. For instance, for neural progenitors, that we, by the way, the neural progenitors, we can isolate them from humans also. This is what we are currently doing with Dr. Akevamins. But the microenvironment, if we put the same type 1 and type 2 in the same microenvironment, one can form neural progenitors, the other cannot. So probably the type 1 don't have some specific receptors for their signals, while type 2 do have. And also in myogenesis, when muscle formation, when we inject the type 1 pericyte, the same number, into the same environment, for some reason they don't form these muscle fibers. So is there a very important factor of the microenvironment? Which are those factors is very interesting, and this I think is the next step of this work, is to try to understand the mechanistics of these processes. Because if we understand what can inhibit and activate at the same time opposite types of pericytes and get the outcome that we want, or that we want to induce in the disease, a drug in the transgenic mice, then maybe we can test the same drug that is specific to the type pericytes in non-transgenic mice. So this is our future goal, is to go deeper into the mechanistics of the environment relation to the stem cells.
ALS, pericytes.
Rabbi?
So,
Wait.
are you aware of any differences in pericytes in models of neurodegeneration, of the role of the individual subtypes?
Okay, so during neuromuscular degeneration or neurodegeneration?
Neurodegeneration.
So neurodegeneration.
So in spinal cord, in ALS, Alzheimer's, Parkinson's, any model.
So in neural degeneration, it is, people have been working with cells very similar to pericytes for years that are the oligodendrocyte progenitors. I'm sorry, so your question is whether the pericytes play a role in neurodegeneration. So pericytes, oligodendrocyte progenitors that are cells exclusive of the central nervous system, have been related with playing many roles and increasing in number after multiple neurodegenerative disease, and as multiple sclerosis, for instance. So they increase in number. However, if you go back to those papers and you analyze, nowadays with the increased amount of markers to really identify a cell, it opens a gate for pericytes. Oh, this could be pericytes. So we have been rereading, doing research, searching again what happened already before, and we have been finding a lot of things like this, that pericytes may be playing roles that before were claimed to be roles of oligodendrocyte progenitors or Schwann cells. Okay. The rabbi question? So the rabbi, I'm sorry if it's finished, the rabbi, he knows very well this story because every Friday I go to his home for Saturday night that we Jews, we celebrate Saturday together, and he knows very well all these stories because usually people from the community ask me what you doing, and then I need to explain. So he knows how to explain very well. He takes cells from the muscle and makes cells from the brain. So, okay, thank you. Appreciate it. Thank you.
There's no like convenient area for one thing, and second, my hours are long, but he's there before. Like because he does experiments.
Yeah.
Sometimes it's in the middle of the night, and I know how he's like computing.
Do you not see him? Because I thought that I was smart and no one take this.
I'll keep repeating this, guys. It's okay, you know.
No.
No, we don't.
No, it's that I'm saying that we have not planned here yet.
Let alone what's going to happen. Oh, okay, I get it.
I just know that I need to go to see her.
You can see, you can tell. Can you tell? Can you tell which one is Mary?
She looks like it.
This is my boss. She bring me some food, she take care that if I'm dressed right, if I'm eating.
You tell me eating everything.
If I'm no, but she is really like my mom was very, 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 I don't know, I felt, I was very like, I felt in the whole lab in general, for some reason I felt like family and all the time. I don't know, many times I asked Osvaldo how can he do the lab this way, and I'm not sure that he knows exactly how it, but for some reason everybody in the lab love each other, and it's very group that like everybody helps each other, everything is so, we are learning every day a lot, and I don't know, it's like an amazing, but you know, usually you come from PhD, you come depressed, or you come already stressed, or it's a PhD. No, it's a normal thing, or people are, oh, phew, I don't have a foo now, I have just one more step. It's like, I don't know, I feel sorry that is a step that I will need to change, something that is already very good. I hope the next thing will be at least see me, you know, at least a little bit maybe close to it, but is something that I just enjoy. I just had a lot of fun in my PhD.
Right. So the philosophy always was in the lab to have a very integrated participation of everybody in research. So it's not as one is the leader of the project, but everybody else helps. So, and I learned that in the hard way, because when I was a postdoc, we were separated and kind of competing with each other. And this is so destructive and counterproductive was that approach. And I thought, when I have my lab, things will be completely different. And in the end, it was more productive, because everybody learned about what everybody else was doing. And the point is that we can't force someone to do everything that is needed for one project, because maybe doesn't have the capacity, the ability, the skills, but someone else in the laboratory can help, and then the project move forward much faster. So,
good luck.
I love that you were part of this story, and you will continue being that part of the story. Thank you. Yeah, I know. Maybe it's a generational problem. Huh? Maybe it was a generation. He said that he had the same experience in NCI. So, people competing in the same laboratory with each other. So let's start eating and enjoying the party.
Alex, do you want to put
Uruguay, Italy?
And so on. He said, let me go back. I will talk to my advisor. I'll decide if I come back. So a few days later, he was already in the lab, ready to start his work. So typical, right? So it's that intensity, he lives his life, and science.
Thank you.
Thank you for working with us. Good.
Enjoy.
Thank you very much for contacting me back, but you know what? I'm in US now. I said, oh, where? I'm in Boston. I said, what? That's a great opportunity for you to come over, visit us, and see what we do. I said, email back and forth. I can say that literally a few hours after he was in the lab. But he explained me that as the time that passed from the first contact was more than some weeks, he already committed. Why?
Because he's like you.
Oh, seriously? I'm not sure that I remember. So when I was a child, my favorite game was like, sometimes I take care of kids. They'll build Lego houses. So some of them, they use gum instead of the good pieces of the Lego. So then I take these ones, I put them in timeout, and I leave only the good pieces. So this is what we are trying to do with PERICYTEs.
I think that's good enough.
Thank you.
So here comes the good one.
That is very good.
Share the Lego.
Okay. And that's to survive in New York.
So this is Laura comes here, right? Come on, because you have been the one that
to recognize that Winston is
everything.
warm weather.
Oh, in serio?
Yeah, but New York is better. That is good. Don't complain about the cold of Winston.
Thanks a lot. So it's very nice.
Oh.
Yeah, to be proud of Wake, yeah. Good.
Nice.
All right.
Very nice.
You know, in my feelings, I can wear it even now. You know, just in case the cold is coming.
Definitely there won't be enough for winter in New York.
No.
So you need to add some layers. Right?
Most definitely.
It's more like me than in the suit, right?
Perfect. And if it's too big, always can be changed.
Thank you.
Nice. Perfect.
Yeah, what?
Because now the boys are with the feet story. The boy with the feet.
This is the...
Feet. The dress are feet.
Oh, the one with the...
No, the feet closed.
Oh, yeah.
Everything is
Slim.
So for that fashion, probably you need to change it. But slim is not.
He's
good.
thin
So
now
the
because
slim
of the
wedding. That's the one. So try both.
Yeah, I don't think it's bad here.
Uh-huh.
He said that.
Yeah.